Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog.
Good evening. I'm Yvonne Stapp for Science for the Public, and I welcome you to tonight's contemporary science program, Looking for Signatures of Life on Mars. To explain how that search is conducted, we have Roger Summons, Professor of Geobiology at the Massachusetts Institute of Technology and a participating science in the NASA Mars Science Laboratory mission. Roger Summons received his Ph.D. in chemistry from the University of Wollongong in Australia, and before joining MIT in 2001, he held appointments at Stanford, Australian Iron and Steel, the Australian National University, and Geoscience Australia. In a remarkable career, he's developed analytical techniques and made discoveries that are today essentials in the field of geobiology. Along the way, he's received many prestigious awards. His distinctions include an appointment as a Fellow of the Royal Society. Dr. Summons is particularly known for his investigations of biological traces in very ancient earth sediments and oil shale. His contributions to the identification of earliest life on our own planet are now applied to the analysis of sediments on Mars. Tonight, Dr. Summons explains how organic matter leaves its signature in the form of biomarkers and how that information can be decoded millions or billions of years later. We are very honored to welcome Roger Summons. Welcome, sir.
Thanks very much.
And I'd like to ask to start with, what's geobiology? What do you do?
Geobiology is the study of how life on this planet has co-evolved with the planet. We know that Earth has changed over geological time, over the 4.5 billion years that it's existed, and a lot of the changes that we know took place on the surface of the Earth have been driven by life. And for most of Earth's history, that life was entirely microbial, that is, green slime. Microscopic organisms have dominated the planet for most of its history.
And are you saying that the geology wouldn't have developed in the same way if it had not been for biology, and maybe vice versa?
Exactly.
Okay.
Exactly.
All right.
One of the most profound changes that people recognise is that the Earth was formed with no oxygen in the atmosphere.
Yeah.
And now we have an atmosphere with 21% oxygen. And we know that oxygen was formed by the photosynthetic organisms that we can see around us, but mostly from the cyanobacteria in the ocean. They were the organisms that invented the ability to do photosynthesis and produce oxygen at the same time.
Right.
And that oxygen has determined how the way rocks have weathered. It's determined the minerals that we can find in rocks. So there's very definite evidence that the activities of microbes have profoundly affected the surface environment and probably the subsurface environment as well.
When we see, say, documentaries or books about fossils, we see these things, sometimes very problematic little fragments and so on, and it is your field and you substantially who developed other ways to trace this early life. You've worked on oil and coal and oil shale and things where we don't think we see fossils. Could you please tell us about it?
Well, the fossil fuel that we're burning to power our cars and our homes is organic matter. It's organic carbon compounds that have been formed in the past by living organisms. All the organic matter, practically all the organic matter we have on the planet, is a consequence of the activity of life. Mm-hmm. So you look at a piece of coal and it's a piece of black stuff.
Yes.
You look at black liquid coming out of an oil well,
right.
it's black stuff.
Right.
But really it's individual molecules. It's individual compounds that we can use chemistry to determine the structures of those molecules. And the structures of those molecules are clearly related to the different organisms that have made the molecules. And those organisms are making their living in different ways. So the structure of a single compound can tell you an awful lot about the organism that made it and the process that organism was using to make a living, whether it was photosynthesis or it was eating something else or eating somebody else. Molecules have a story to tell, and that's what I've been very fortunate to be able to work on for the last 40 years of my career.
So it's this combination of chemistry and biology and I don't know what
geology.
else, exactly, all of that together. But again, I think most of us are just not aware that you could look at oil and coal and find a history like this. And so you do this sort of chemically with looking for markers. Is that the idea of traces?
We use a technique called gas chromatography combined with mass spectrometry. And to explain that a bit more, gas chromatography is a way you can separate molecules with incredibly high resolution. So you can separate molecules that only have very, very subtle differences in chemical structure. And the mass spectrometry part is an approach to getting a fingerprint of those molecules. And we can compare that fingerprint with molecules that we can make in the laboratory or molecules you can buy off the shelf from somebody. So it's a lot of comparison. Sometimes we find molecules in rocks that we've never seen before. And in that case, we have to do a lot of chemical experiments to determine the structure. Once we've determined the structure, then we have a sort of a chain of connections that we can make to through biochemistry, basically, to the organisms that made those compounds.
When the rover drills a hole in a rock or in the soil in Mars, you can really look at this. You don't have to see the fragments in a rock. As we are usually shown with fossils when we think of fossils.
Yeah.
So.
The best way to think about the MSL mission, I think, is think of it as a geologist, a robotic geologist on wheels.
Yes.
And the MSL, or the Curiosity rover, has a whole suite of tools that a geologist would normally have. For example, it has a hand lens.
Yes.
And the hand lens produces beautiful photographs. It's just the most spectacular instrument.
Takes little selfies.
It takes selfies and takes mosaics.
That's
it's
amazing.
a brilliant camera.
Yeah,
and were there microscopic organisms on Mars, that hand lens would be able to see them. I mean, they would need to be a certain size before it could resolve them from the rest of the rock. But with a hand lens, you could see a moderately sized small fossil if they existed.
That's amazing.
Well, I've got a cartoon of the rover,
Okay.
and the cartoon explains what the primary mission is. The primary mission is not about detecting life. The primary mission is about determining whether life could have existed in the past at some place on Mars. We can't explore the whole of Mars.
Obviously.
It's very, very difficult.
Right.
But there was a very detailed selection process that chose a place where they thought they knew a significant amount of the geology. There's no point going to a place where you have no context, because in this sort of work, context is everything. So a lot of selection went on to choose a place where they thought they would have rocks that could preserve life signatures had they been formed at the time.
Right, I see.
And as a consequence, they chose Gale Crater as the landing site. And within Gale Crater, then the tasks that Curiosity was charged to perform, Curiosity and of course the team of scientists that runs the mission, is to evaluate the biological potential of that place, to understand the geology and geochemistry and their interrelationships, particularly to try to understand the role of water in that particular environment. And because life is profoundly affected by radiation, both beneficially and deleteriously. If we get too much solar radiation, that's not good. So evaluating potential for life to exist really depends on understanding the entire environment, including the radiation environment. So that's the mission, not to detect life, but to see if life could have existed. This is another cartoon of curiosity, and it's got the various instruments labelled, such as mast cam, chem cam, Marty. These are all imaging systems. So I think there's altogether 9 cameras. So the mast cam takes big images, it's stereoscopic and colour images. There are hazard cameras that on the wheels that the rover drivers can use to evaluate danger, and it also shows the instruments that are inside the rover. The main chemistry instruments. There are 2 of them. One of them is called SAM, Sample Analysis on Mars, and that's the experiment that I was selected to work on. And then another one called ChemIn, which is an X-ray instrument that can determine the minerals that are present. And the minerals that are present give you a lot of context. So if you get the information about what minerals, quartz, olivine, carbonate, those sorts of things, it helps you evaluate the information that you're going to get from SAM. SAM collects data on soil samples, on sediment that would be drilled by the drill. SAM also has the capability of looking at the gases in the atmosphere. The gases in the atmosphere comprise carbon dioxide, water vapour, a little bit of oxygen, argon, some nitrogen, and those gases and their isotopic compositions can tell you an awful lot about the history of the atmosphere. So the mission is not just about looking for organic molecules, but evaluating the context of the samples that we're analysing. It's an ongoing evaluation process. They're using data that's collected by the rover itself. They're also using data, imagery from satellites that are orbiting Mars and taking high-resolution images of the surface of the planet. So it's a combination of information on the ground using the cameras and information from above using the satellites to place the rover in a position where they might find an interesting rock. And there's no point in looking at any old rock. We're interested in those rocks that best preserve organic matter. And on Earth, the typical place we would look would be in a shale that is a very fine-grained sedimentary rock. We don't find organic matter typically in a basalt or a granite or an igneous rock. And we don't find it in a sandstone because the grains are so big, anything that was in there would leak out. It's very fine, muddy, silty sediments that best preserve organic matter. Also laminated sediments, layered sediments that have been put down over a period of time. If the cameras can show us a layered sediment, that's another indication that the sediment was deposited underwater. It's one of the main factors that we look for on Earth to find organic matter, whether we're doing it to find coal or oil or whatever. It's no use looking in sand in the Sahara Desert that's exposed to the atmosphere that wasn't deposited underwater. We're looking for fine-grained sediments deposited underwater that stayed or had low concentrations of oxygen. If you've got a lot of oxygen in the environment, organic matter is good food for bacteria. So it would be consumed, eaten, oxidised, it would ultimately disappear. So we're looking for places where organic carbon and other signatures are preserved. It's called the science of taphonomy. It's a long word. Taphonomy is understanding all the processes that lead to the preservation of a fossil, whether it's a molecule or a microbe or a bone or a human skull. Taphonomy is the science of understanding where best to look for well-preserved material. And we apply the science of taphonomy to looking for the ideal places on Mars where something useful or interesting might be preserved. So there's an ongoing process. Every day, members of the science team are looking at the images, evaluating where the rover is, evaluating targets to look at in the future, and then they develop a list of priorities. Those priorities are uploaded to Curiosity, and it goes through its steps, doing the experiments one after the other that the science team have determined is going to optimize the data that they're going to get down. So it's ongoing. Well, when the mission first started, it was 24 hours a day. Now they've gone down to 5 days a week, so 12 hours a day.
A couple of days off for the...
A couple of days off for Curiosity and for the scientists.
Yes, I guess so. You have written about the difficulty, I think you just mentioned it also, on a planet like Mars. Radiation is a really serious destroyer. So how do you get around that? First of all, I think you mentioned there's not really an atmosphere like we think of atmosphere now, so it would be, is it worse?
It's an atmosphere that's very thin. And Earth has an atmosphere we call one atmosphere. Mars has an atmosphere about 1% of Earth's atmosphere.
Right,
So for every 100 molecules in Earth's atmosphere, there's one molecule on Mars. And that atmosphere is not dense enough to stop or to inhibit the radiation.
Okay.
And consequently, the surface of Mars is getting a full dose of solar radiation.
Right. So
even though Mars is a little bit further away from the Sun than Earth, that radiation is not stopped by the atmosphere.
Okay, so that's one thing, so that you would have to go internally. So you pick a rock and you know you have to dig for this, correct?
Yes.
When you get the samples, and the project is sending you the data, sending this enormous team of people all this data, you have machines in your lab to do, I think you said, analog kinds of experiments. Could you tell us? So here's some data that looks interesting.
Yes, it's part of the process of ground truthing. Ground truth. Yes, and all science relies on ground truth. You have a hypothesis, you do an experiment, and then you query that experiment. You try to find fault with it. Your job is to disprove yourself. And so there are many people on the SAM team doing these experiments daily, not just at MIT, but there's a large group of people at the Goddard Space Flight Center in Greenbelt, Maryland. There's people in Houston, at Johnson Space Center, all doing analog experiments and testing various aspects of the results that are coming down. And one of the things we do is make a mineral or a mixture of minerals that we call an analog soil, for example, or an analog sediment. We take the information that we've got from the rover about the mineral composition, and we make a similar mixture in our lab, and we run the experiment as close as we can to the way it's been run on Mars. And it's those experiments then determine or inform how the experiment might be done on Mars better than it was last time. So for any rock that we look at on Mars, we might look at it, do 6 or 7 experiments based on a combination of the results that came from Mars, but also the results that came from Earth. There's actually a SAM instrument, testbed instrument, that it's operating under Mars atmospheric conditions in a chamber at Goddard Space Flight Center. And that's the main place where analog experiments are done, using an instrument that's almost identical to the one that was flown And that way they can test the software, they can test many, many more aspects of how the experiment works and check them for truth.
Yeah.
And it's a constant backwards and forwards of experiment on Mars, some experiments on Earth, redo the ones on Mars, redo the ones on Earth, and you're finessing your understanding of the system.
Right, and obviously a lot of cross-checking among the scientists themselves, correct?
Yes.
So that's one of the most interesting things that I read about your work with looking at fossil life is that DNA and other proteins don't preserve well, and certainly would not do on Mars.
Yes.
Then you look at lipids. Can you tell us about this and what's lipid?
What's lipid? This picture shows some chemicals that we might find on Mars. Okay, and on the top row are very, very simple molecules. Acetic acid, vinegar. There's a straight-chained hydrocarbon, simple hydrocarbon that you might find in gasoline, for example. And then the compounds with the rings called polyaromatic hydrocarbons. And those sorts of molecules can be made by all sorts of processes, not necessarily involving biology.
Okay.
They are formed through biological activity, but they're not diagnostic for biological activity.
I see.
We find those molecules, for example, in meteorites that land on the Earth, or in interplanetary dust that's always landing on the Earth. So they're not particularly informative about the processes that made them. Now, the second row of molecules are typical of the structures that are formed by biology. And on the left, we have amino acids, and the one shown there is alanine, so it's part of our proteins, for example.
Of that 20 that make up life here on Earth?
It's one of the 20.
Okay.
Has another feature to it, and that is a handedness.
Aha.
What we call homochirality, and all the amino acids have the same configuration. And that's a very, very different thing to the sort of nondescript, or not, that's a bad word, but the less informative characteristics of the molecules on the top row. The molecules on the bottom row have structural complexity that can only be imparted by biology.
Okay.
And the molecule on the right, large complex molecules with three-dimensional qualities, and the one I'm showing there is cholesterol. And we wouldn't be here without cholesterol.
Yes.
Cholesterol is essential to making our membranes work. It's a very complex molecule, and it's made by a complex series of reactions in our cells, in biochemical or biosynthetic reactions.
And this is a lipid as opposed to the protein. Is that correct?
The molecules on the left, amino acids,
okay,
because they have the chemistry makes those amino acids rather unstable over billion-year timescales. They're stable over our lifetime.
Yes.
And we can find them in fossil humans.
Right.
From Olduvai Gorge, for example, places where people are looking for early hominids. They're stable over that sort of timescale, but they're not stable over billion-year timescales, which is what we're looking at Mars, because Gale Crater, we think, was formed about 3.5 billion years ago, 3,500 million. That's a lot different to a human timescale.
And also that it would be true, is it for Earth, when you are looking back 3 billion years on Earth at what you hope are fossils or something, you emphasize these lipids as opposed to the protein, right? The protein, I just need to understand, doesn't last so well.
Protein, DNA, sugars,
nucleotides, and
all of those sorts of molecules, they're unstable for several reasons. There's a chemical reason, that they have what we call functional groups that are easily attacked by bacteria. But they're also good to eat because they have nitrogen and phosphorus, and they have elements in them that microbes like to eat. So if you've found an amino acid in a very old rock, you'd have to be really suspicious. Why didn't someone eat it in the billions of years that have gone on?
I see.
But it's much, much more difficult to eat a molecule like cholesterol. I often use the example that when we're dead and buried and we sit in a coffin for, I don't know, a couple of 100 years, most of our biomass will have been consumed. The thing that of us, or the part of us that's the most recalcitrant type of material, are our lipids. So our cholesterol is going to last a lot longer than we do.
That is quite interesting, yeah. And so therefore that's what you're looking for.
We're looking for
on Mars
molecules
too. Right,
that
like,
are very difficult for
right.
other organisms to eat.
But they would still clearly signify biological, or I don't know if prebiotic, but something along those lines, right? Not.
Yeah, they would preserve the qualities that we associate with biology.
All right. That is quite interesting. Has that gone on for a long time, and the rest of us just don't know it, that you've known this about fossil residuals anywhere?
The field really took off. Well, it was started in the 40s and 50s in a little way.
Yeah.
A gentleman by the name of Alfred Tribes identified a molecule in a rock that he could relate to chlorophyll. And of course, chlorophyll, we wouldn't be here without chlorophyll.
Right, yeah.
And identifying a molecule, it's called a porphyrin, which has got the centre of a chlorophyll, or that's found in the centre of a chlorophyll, clearly tells you that the organic matter has something to do with biology. And then for the next 50 years, people explored that idea in greater and greater detail, and you can ultimately show that every molecule in every drop of oil on this planet was formed by life at some time in the past. Not only that, the type of life on Earth has evolved over time.
Yeah.
We started out with mostly bacteria. Those bacteria, or parts of them, were incorporated in more complex organisms to form algae.
Yeah.
Those algae evolved into more complex organisms we call plants. And that process of evolution can be traced in oil. The composition of oil has also changed over geological time, corresponding to the way the plankton in the ocean has changed over geological time. So
it's a very faithful marker of the evolutionary process.
And it's totally complementary to the fossil record, the visible fossil record, or the microfossil record. So we have colleagues who study microscopic organisms in sediments, and they can see the evolution in plankton through time. And just the same as they see rocks dominated by cyanobacteria, for example, and later at some later time rocks with green algae, and then at some later time more complex algae like diatoms and dinoflagellates. You can see those transitions in the fossil record, but you can also see those transitions in the molecular record.
Which is amazing. Yeah, that's because that's a very, very accurate, I guess. Kind of markers.
Accurate. It makes sense too.
Yes.
I mean, all science is about making sense of the environment around you and making sense of the phenomena we experience. And we live on a very, very complex planet, and it is very complex, largely to do with the presence of life.
Yeah.
And the history of Earth is very, very different now, we now know, to the history of Mars. Largely as a consequence of Mars being smaller than the Earth, and Mars having lost its water. It may well have had as much water in the beginning as Earth had, but that radiation from the sun and the smallness of the planet means it lost its heat more quickly than the Earth did, and didn't develop the cycles that the Earth has developed that help us preserve the water. The water in the atmosphere are what's blanketing us and protecting us in a large degree from solar radiation, as well as the magnetic field. Mars probably had a magnetic field very early on, but also lost that. So it lost the ingredients that would have made the planet habitable. We know that. What we're trying to do is understand something about the very early history of Mars, and when it still might have had a magnetic field and might have had an atmosphere, might have had an ocean. And that's the sort of logic that curiosity is trying to develop. And of course, curiosity is just one theme in a long history of exploration of planets, largely driven by NASA, but increasingly by ESA, the European Space Agency, and now China and India.
Right. Everybody will be in on
Everybody's
this.
in the game. But it's all about sharing knowledge.
Yes, and that's the other thing, the sort of a real community, maybe peace will break out or something in this community, because there seems to be a fair amount of sharing with.
I think science, like arts and music, they're ways of soft diplomacy.
Yes,
and
exactly,
that's why
but pretty effective.
well, that's why I love being a scientist, because you can interact with people in any country in the world on common ground, and we're not concerned about our differences, we're concerned about our common ideals and our common hopes. And I think it's one of the great joys of being a scientist, and it's why I would recommend that science as a career for young people.
Well said. Back to Mars for a minute. There has been this controversy around methane. I don't know whether it's controversy, but that methane can be, they're always careful with the modal, can be an indicator of life, but it isn't necessarily. Can you explain that to us? And then in the last several weeks, sometime in the last several weeks, that was sort of confirmed, a methane-like cycle on Mars. What do you think of that? And first of all, though, about the methane itself.
So now I'm showing a picture of SAM.
Okay.
And the SAM instrument is really three instruments. It's a gas chromatograph, which I explained is used to separate molecules. It's a quadrupole mass spectrometer, which is used to identify those molecules. And it's a tunable laser spectrometer, TLS is the acronym, that can look at gases, and it can measure the spectrum of gases. And it's been used to largely to evaluate the atmosphere, but if we heat a soil up or crushed up rock and heat that in the system, the gases that are evolved can be looked at in the TLS. And of course there had been reports of methane on Mars in the past based on information from telescopes on Earth, as well as orbiting telescopes, or spectral combination of telescope with a spectrometer.
So it has this equipment, it can identify, and presumably it was instrumental in determining this thing. But what is the issue then with the methane?
Well, the issue with the methane is that it had been detected from Earth, but when you look through Earth's atmosphere, there's methane in our atmosphere. So the difference between the signal they are looking for is teeny weeny on top of a much larger signal from our own atmosphere. So if you're using a telescope
I see,
on Earth, you're hoping to see through the atmosphere, but there will be methane in that, and see a little bit more signal when you look at a particular place on Mars. So there was a lot of controversy about how real was the little signal when it sits on top of a much bigger, noisy signal. And being able to measure it in situ in Mars' atmosphere on the surface of Mars was considered to be a much more reliable measurement. And so the instrument takes in a sample of atmospheric gas, freezes out the carbon dioxide and some of the other components to enrich the sample. And ultimately they've made, I think, 4 measurements where methane is just a little bit above the background of the instrument. So in all of this work, you're looking for a signal on top of inevitable background.
I see.
We call it signal to noise ratio.
Okay.
And the signal that they're getting from methane on Mars is very tiny. It works out to be about 7 parts per billion, so it's minuscule compared to the amount of methane that's in the air we're breathing.
Okay.
Very small amount.
All right.
And not only that, it was not there in the early part of the mission. The rover got to a certain place and suddenly they made 4 or 5 measurements where they got this small amount, and just as quickly as it appeared, it disappeared. And the appearance and the disappearance of the methane does not correlate with any other measurements that Curiosity's made. It doesn't correlate with day-night. It doesn't appear to correlate with the amount of radiation. Doesn't appear to correlate with what month of the year. There's no other measurement that Curiosity's made that bears any relationship to the appearance and the disappearance of the methane. So that bothers people. You should, if it comes and it goes, you should be able to understand why that is. But that was the same as the experiments when they detected it using Earth-based satellites, Earth-based telescopes. Sorry.
But that was interference. Yeah.
Well, it appeared and it disappeared.
I see. Yes.
And so it appears that it's coming from somewhere, but in very small whiffs.
I see.
And there's no really solid understanding of what those whiffs mean.
Okay.
On Earth, a lot of the methane comes from biological processes.
Right.
And those processes overwhelm the formation of methane from non-biological processes.
Okay.
And so even though you can identify a small amount in Mars' atmosphere, there's no way there's enough of it to get any other information that would tell us whether this is biological or non-biological.
Okay.
So it's a really interesting and intriguing scientific problem, but it's also an enigma.
At this point.
At this point.
Right, so you're not speculating on it even.
There are many aspects of the behaviour of materials on Mars that are different to Earth. One of the things is Mars' surface environment is very oxidizing. Over its history, the water has been what we call photolysed to hydrogen and oxygen. So H2O goes to H2 and O, or O2. And the hydrogen, because the gravity is lower, the hydrogen can be lost to space, leaving the surface enriched in oxygen. And that oxygen is mostly tied up in minerals like sulfates and perchlorates, strongly oxidizing minerals. And strictly speaking, the processes that form those minerals should also destroy the methane. So the fact that there's some methane still there tells us that there must, if it's true, if the measurement is real, that there must be some process still generating the methane, and that is even more intriguing. Some people would like to think it's biology, but there's no evidence. Of course,
everybody wants to think it's biology. I have to let you talk to the audience here very soon, but is there anything that you would speculate on about Mars in terms of this project that you'd like to tell us about?
It's proved very, very difficult to find any organic matter on the planet. And organic matter on the Earth is a biosignature, and the failure to find organic matter on Mars might be interpreted as the absence of a biosignature. But again, we think it's a strong radiation environment and the presence of these oxidizing materials like sulfate and perchlorate and oxygen itself that's destroyed it. So the drill can only go in a couple of centimetres. We can't go deep enough that where you might find material that's been separated from these nasty processes.
A lot of people want to have life under the surface of something or other. You know, I remember Thomas Gold, as a scientist here, speculating that the Earth very deep down was full of bacterial life, you know.
It is.
And evidently it turned out, but he was laughed out of the room when he first mentioned that, I think.
Yeah, but he also said that fossil fuels aren't formed by biological processes.
Yes, that's right. All right, I'll go there.
So he was right about the deep biosphere.
And very wrong about a few other things. But in any case, people want to find life out there in the worst way. Do scientists sort of, are they optimistic, pessimistic, or is just totally bewildered, like it's too hard to reconstruct?
Not so bewildered. It's taken hundreds of years for us as scientists and particularly geologists to understand and put together some history of the Earth. It required all sorts of scientific inputs, like the discovery of radioactivity gave us a tool to date rocks precisely. People used to estimate before they had real dates from uranium-lead decomposition. It's a very long, iterative process to understand the history of the Earth, paleontology, geology, geophysics.
And now you need all of it, it seems like.
And that same series of streams of logic and knowledge are being applied to Mars. But what we're finding out is Mars is very, very different in all sorts of interesting ways. And ultimately people will come to an understanding of those processes through this mission, and hopefully it continues for a few more years. The rover stays alive, and
it's amazing. This machinery by itself is,
and it speaks in a spectacular way to the engineering skills of all the people that worked on developing the rover itself, the delivery system, the landing system. It's just a spectacular...
It's a real tribute to humanity, I must say, isn't it?
Yeah. And a lot of those scientists are US-based, but there are also a lot of them based in other countries. There's Russian scientists and there's French scientists working on this mission. So it really is international, and it really is a spectacular scientific and engineering accomplishment. There's one other important aspect, and that's leadership. The leadership of the mission has been spectacular, and the principal scientist, John Grotzinger, who used to be a professor at MIT, led that mission for the 7 years, the most critical 7 years. And I think his way of getting people to work together and be critical of each other without
in a
cutting each other.
Positive way, a productive way, I should say.
The whole job, your whole job there is to do good work, but also to prove someone else wrong. And to get people to do that in a constructive way is a remarkable achievement.
So the whole mission is spectacular. I have to leave it there so that you can talk to the audience. Dr. Summons, thank you ever so much for being with us.
Thank you for having me. And